Hello, everybody. I'm really pleased to have today Eric Vivier with us, thanks to this event organized by the International Union of Immunological Societies. Eric Vivier joined the Aix-Marseille University as a professor at the Centre d'Immunologie de Marseille-Luminy in 1993, where he became the director from 2008 to 2017. He is currently the scientific director of the biotech company called Innate Pharma. Eric Vivier has made several scientific contributions, especially in the field of innate immunity, where he used several transgenic mouse as a model to study the ontogeny, the function, but also the manipulation or the therapeutic manipulation of NK cells. Eric Vivier has also been involved in development of innovative treatments for cancer. And more recently, he has also made major contributions in the identification and characterization of the innate lymphoid cells. He has over 370 publications, and he is also a member of the French Academy of Science, to really name few of his contributions. Thank you very much, Eric Vivier, for being here with us. We're really delighted and looking forward to hearing your talk. Please go ahead, Eric Vivier.
Okay, thank you, Abdallah.
Uh,
Thank you very much for the
if,
kind
I, if,
introduction.
if I may just add to the attendees, they can ask questions. They can put the questions whenever they want. I will be, collecting questions. Go ahead. Sorry, Eric.
No worries, Abdallah. Thank you very much for the kind introduction. Thank you actually to the IUIS, and thank you also to Frontiers for this very kind invitation. So it's really my pleasure today to share with you our very recent results. They are all unpublished as of today on the association of COVID-19 inflammation with activation of the complement pathway, and in particular with the activation of the C5a-C5aR1 axis. So this is my disclosure slide. So as Abdallah said, I am a scientific officer and chief at Innate Pharma, but also a professor at the Marseille University and working as the Center for Immunology in Marseille-Luminy. Okay, so what we've been doing facing the emergency of the COVID pandemic is actually to gather a group of very motivated people, comprising people from the Marseille Public Hospital, people from Innate Pharma, and people also from the Center for Immunology in Marseille-Luminy, in a federation under the auspice of Marseille Immunopole, which gathers all the people working in immunology in Marseille, to try to understand what's going on in terms of the immune response in the presence of SARS-CoV-2 infection. So what we know is that most COVID-19 patients present only a few mild symptoms, but a few percentage of them, 15% roughly, progress to severe pneumonia, and about 5% develop acute respiratory distress syndrome, that I will call ARDS for the remaining of this presentation. And these people having pneumonia or ARDS have to get some therapeutic strategies, and these ones are urgently needed. So we know that the immune system plays a dual role in COVID-19. It's obvious that the immune system is contributing to virus elimination in the first phase of this disease, but it also contributes to ARDS development, as we will see. So we, as I said, we set up this large translational study called Explore COVID-19 with the aim of characterizing with some depth, obviously, the immune response occurring during disease progression from the mild to severe forms, and I will update you on that in a minute with a list of patients that we have enrolled in our study. And the idea behind that was actually to evaluate whether it was possible to repurpose some drugs which are already available, either because they are approved or because they are available in clinical trials for fighting this severe lung infection. So this is quite a busy slide. I hope that you all can see on the right-hand side the details on the cohort of patients that we have enrolled. But basically, we have been working in a longitudinal manner with 3 different time points. The inclusion time, then a T1 which is basically day 4, and another time point which is called T2, which is day 10 after enrollment. And so, as you can see here, we had 10 healthy controls, 10 paucisymptomatic COVID-19 patients. All the data will be in blue for this cohort of patients. Patients with pneumonia, but not ARDS, will be in green, and there were 34 in our study, and 28 patients were included with ARDS, and they will be in red for this presentation. So some obvious features to begin with. We could see that there was indeed a seroconversion in some of these patients. Actually, not all of them. You can appreciate that the seroconversion was readily detectable in patients with pneumonia in green or with ARDS in red, but it was much lower, the detection of antibody for patients with paucisymptomatic symptoms. Now, we had the chance to collaborate with our colleagues in Marseille, who are looking at not only at the antibodies against SARS-CoV-2, but also on the seroneutralization properties of these antibodies. And as you can see, one could really detect seroneutralizing antibodies in almost all, actually all ARDS patients, most of pneumonia patients, and a substantial fraction of paucisymptomatic patients. So these results really recapitulate what we know about the antibodies which are raised during SARS-CoV-2 infection. Now let's look at the lymphoid compartment, and we could also here confirm that COVID-19 is associated with a severe lymphopenia, which associate NK cells, CD8 T cells, CD4 T cells, and B cells. And as you can see, this lymphopenia is somewhat commensurate to the severity of disease, and those results, this part of the results regarding the immunomonitoring of the lymphoid compartment in press in cellular and molecular immunology will be available very soon. But we were more concentrated on other aspects, in particular the myeloid cell compartment and the complement cascade. But before going into that, I would like also to show you that we here also recapitulate what we know about the famous cytokine storm which occurs during COVID-19, in particular in patients with ARDS. And you can see by looking at IL-6, CCL4, CCL2, or CXCL9, this is indeed the case. We also monitored the C-reactive protein, which is very important. And you can see that this is following basically the same kind of kinetics, so to speak. So, but we focused on the complement, as I told you from the title of this presentation. Why did we focus on the complement? Well, the complement is obviously very important in terms of immunity, in particular immunity against bacterial infection, not only, but particularly. But there is also another part of the complement cascade which is involved in inflammation. And as we were facing an inflammatory disease, it was quite obvious for us that we should include the monitoring of the complement cascade. But more than that, as I told you in the introduction, as our aim was also to find out whether it was possible to repurpose, to reposition in the context of COVID-19 some molecules, some drugs which are already available. And as Innate Pharma is developing an antibody that blocks the C5a-C5aR pathway, it was obvious for us to focus on this pathway. So I remind you that there are 3 pathways to activate the complement. You can really appreciate here in red, orange, that the C5 is really a critical part of the activation of the complement. And I remind you that when C5 is cleaved into C5a, which was also known as an anaphylatoxin. It's a very pro-inflammatory molecule, whereas C5b associate with the other component of the complement cascade, the late component of the complement cascade, namely C6, C7, C8, C9, to form the MAC or the soluble deposit of C5b9, and we'll come back to that. So our study was actually supported by earlier data obtained by this group in China, where these colleagues have actually shown that the N protein of SARS-CoV-2, but also SARS-CoV and also MERS-CoV, the other coronavirus, actually bind to MASP2. So as you can see on the right-hand side of this slide, MASP2 is a key serine protease which is involved in the lectin pathway of the complement activation. And this leads to overproduction of C5. And these colleagues already claimed at this period of time, so, in March, that C5 could be involved in inflammatory lung injury. So there are also other data, not with SARS-CoV-2, but with other viruses, in particular with viral-induced pneumonia and ARDS, that show that indeed C5a could be a critical factor in the in this inflammation. So as you can see here in the mouse with H1N1 or with H5N1 or in non-human primates with H7N9, you can see that at each time the blockade of the C5a pathway is actually very beneficial in these animal models. And you can see here on the right-hand side, I hope that you can appreciate what's going on here. Those are non-human primates infected with H7N9 in absence of C5a antagonist antibody. And you can see infiltration in the lung of macrophages and neutrophils. But you can appreciate that when the C5a pathway is blocked, you have actually a clearance of this infiltration, which is so important. So based on that, we have actually analyzed in our own cohorts of patients what was going on regarding C5a. So as you can see here, there is a very impressive correlation between the level of circulating C5a and the severity of the disease, from paucisymptomatic to pneumonia to ARDS patient. I would like to mention that this is happening prior to cytokine storm, as if you recall the slide that I've been showing you before, in paucisymptomatic patients, you don't have much of circulating cytokines. And the second point also is that although IL-6 has been very well documented as being an important parameter, an important factor in the development of ARDS, in our cohort of patients, we had some ARDS patients with no circulating IL-6 detectable, which is absolutely not the case for C5a. So this result supports the fact that C5a is not only a biomarker of the severity of COVID-19 pneumonia, but it could also be causative, and this is what we are going to explore together. So importantly, our translational study was a longitudinal study, as I told you, namely that we have followed the 72 patients over 3 time points, as I told you, day 4 and day 10 after inclusion. And what is remarkable is that the level of C5a remains stable for most, if not all, the patients. So what we could see also as a marker of the activation of the complement cascade is a deposit of C5b9. So the association of C5b with the other late component of the complement cascade in this very interesting lesion that can be seen in the lung of COVID-19 patients, where we could see obliterating under arteries, meaning that this is an arteries, and you see that it's totally blocked by infiltration by several kind of cells. And as you will see, these cells include cells expressing the C5a receptor, but at this stage, what we can detect is a deposit of C5b9 that illustrates again that the complement cascade is really there and may be playing an important role in the onset and the maintenance of the inflammation. So this is for C5a. So now what about C5a receptor? So there are two kinds of C5a receptors, C5aR1, which is very well known as being pro-inflammatory, and C5a-R2, which is here, which is less documented in terms of biological function, but there is no evidence that it could play any role in inflammation. It's rather the other way around. So we focused on C5aR1. So as you can see here, C5aR1 is really highly expressed on myeloid cells. This is true in a healthy individual, where 100%, for example, here of neutrophils in panel C do express C5aR1. This is also true for monocytes, where usually more than 85% of circulating monocytes express C5aR1. Now, what you could see during the course of COVID-19, panel A and panel B, is that in panel A, there is a neutrophilia, which has been well described by others in the context of COVID-19. We didn't see any major change in the number of circulating monocytes, but what we could see when we split monocytes into conventional monocytes and so-called inflammatory monocytes, CD14 low, CD16 plus, one sees that these cells are disappearing from the circulation, and you will see, but I can anticipate the result, that this is maybe the consequence of homing of the circulating monocytes from the circulation to the inflamed lung. So not only the expression of C5aR1 is high on myeloid cells, neutrophils and monocytes, but it's also very stable. I remind you the color code. Green is pneumonia and red is ARDS, and you can see that the expression remains stable along the disease, the course of the disease. So now, what is C5a doing? So what we've been doing here is actually to take PBMCs from COVID-19 patients and activating them overnight with LPS or AR848. I remind you that AR848 activates the TLR7, TLR8 pathway, and TLR7, TLR8 is a pathway that can detect single-strand RNA viruses, such as coronaviruses. So we do think it's good to use them in vitro, and we could actually stimulate these PBMCs in the presence or absence of C5a, in addition to LPS or AR848. And as you can see, first, there is an induction of inflammatory cytokines by LPS and R418, which is of no news, but there is also an increase in IL-6, TNF, and CSCL2 production induced by C5a in this in vitro system. So that was for the blood. Now, what about the lung? So we've been able to collect broncho-alveolar lavage fluids from a fraction of COVID-19 patients in ARDS. And as you could see, and very importantly, we can detect C5a in this broncho-alveolar fluid, but we could also detect inflammatory cytokines. Not much of IL-6, by the way, but CXCL9 was there, CCL2 was there. Importantly, CXCL8, which, as I remind you, was also called as IL-8, which is a very important factor for the chemoattraction of neutrophils. All those molecules are in the BALF. Now, when we look at what kind of cells are there, we can detect myeloid cells, and we can detect myeloid cells that express C5aR1, such as is in neutrophils and monocytes. So all the factors are there. C5a is in the blood. C5a is in the bronchoalveolar lavage. You have also cells, myeloid cells, that express the C5a receptors. They are in the blood and they are also in the bronchoalveolar lavage. So all the factors are there to find out that maybe this C5a-C5aR1 pathway is very important. But let's look at the tissue in more detail. And here we've been using multiplex digital pathology to look at the expression of C5aR1 in combination with CD68 or CD163 macrophage marker in control lung, non-COVID, or COVID-19 lung samples. And as you can see here, it's very obvious there is an infiltration of cells of the myeloid lineage expressing macrophage slash monocytic markers, but also co-expressing C5aR1 for a large fraction of them. As you can see here, there is an obvious difference between control lung and COVID-19 lung. So really, all the actors are there. And we've been completing this kind of analysis by looking again at these very specific obliterating endarteritis lesions, where you can see here again that the artery is totally blocked by this infiltration of cells. And you can see that these cells, some of these cells are of myeloid origin, and a fraction of these cells express C5aR1 as well. I remind you that there is also a deposit of C5b9 in this lesion, as I've been showing you. So now we have the chance to reanalyze RNA-seq data, both bulk RNA-seq and single-cell RNA-seq that had been published by other groups, in particular in China, and made available, and thanks to these colleagues. And as you can see, we can confirm at the RNA level the signature that we could see at the proteomic level using this kind of analysis, where you see that macrophage slash monocytic signature is really increased in COVID patients as compared to healthy donors. The same thing for the complement pathway. And this is true for PBMCs, but it is also true in particular for the myeloid signature in the lung of COVID-19 patients. So as I told you, we could also have access to single-cell RNA-seq data. So we could reanalyze that in using classical UMAP projection. But we've been using Harmony, which is a very nice software suite that can reanalyze these different subsets, and we can really detect myeloid cells in the lung of COVID-19 patients, as well as other cells, but I will focus on myeloid cells for obvious reasons. And basically now when you look at this single-cell RNA-seq data, you can really identify subset of myeloid cells that basically integrate different clusters. And you can see that in healthy controls and in ARDS, there is a major change in this myeloid population infiltrating the lung. And when we look at usual suspects, such as CXCL8 again, CXCL9, CCL2, CCL4, I'm sorry, the dots are a little bit small, but I think that you can appreciate that there is much more, in fact, of these pro-inflammatory cytokine genes expressed in the context of this myeloid cell subset in the BALF of ARDS COVID-19 patients as compared to healthy control. Interestingly, C5aR1 is expressed on myeloid cells on both sides, meaning healthy control and ARDS patients with COVID-19, which really tells us that the critical factor for an inflammation due to the C5a, C5aR1 is really the production of C5a, as C5aR1 is expressed at steady state in these myeloid cells. So now, what does C5a? I've been telling you that it was stimulating the production of cytokines, but it does something else. And so the purpose of this slide will be to show you what it does, both in vitro and in vivo in a humanized mouse model, and also to tell you that we have tools or even drugs to manipulate the pathway. And I will be focusing on one antibody, a therapeutic antibody called avdoralimab, which is a human Fc-silent anti-C5aR1 antibody, which is actually developed in immuno-oncology by Innate Pharma, then in the context of cancer. So the first set of data is represented here. So what you see here is actually the induction of CD11b, on freshly isolated neutrophils induced by C5a. So basically, you start from here in gray, and you see the induction of CD11b induced by C5a in vitro. What you can appreciate is that when you treat the cells in the presence of avdoralimab, the anti-C5aR1, you totally block the induction of CD11b. And you can see that over a dose response of C5a, just to show you in purple that avdoralimab, this anti-C5aR1 blocking antibody, is really extremely potent. And now we could compare actually avdoralimab with other molecules that can impact on the pathway. So we had access to an anti-C5a monoclonal antibody, or we had access also to avacopan, which is an antagonist of the C5aR1. And you can see that each time is the same. By blocking the C5aR1 receptor, one blocks the activation of neutrophils induced by C5a. But now, something which I think is key is the fact that C5a is inducing the migration of neutrophils. This is obviously of interest when we see the infiltration of myeloid cells in the inflamed lung of COVID-19 patients, and when we know, as I've been showing you, that C5a is present on site in the broncho-alveolar lavage fluid, but also looking at the C5b-C9 deposit. And so what we have been doing here is to take freshly isolated neutrophils in a very simple transwell assay and to see that indeed C5a is inducing the migration of neutrophils. And this is totally blocked using the anti-C5aR1 antibody, which I think is very good news for the possibility of using this molecule in COVID-19 patients. But we wanted to challenge actually this in vitro data, although they were obtained with human cells, which is obviously an advantage, by adding another model using this humanized mouse. So this mouse was humanized for a single gene, which is C5aR1 by itself. So we've been using a knockin mouse described by others, and thanks for sharing the mice, that express C5aR1 of human origin instead of mAbs C5aR1. And what you can see here is that in the knockin mouse, there is actually a quite nice expression of human C5aR1 on neutrophils and monocytes that clearly mimics exactly what you have with the endogenous expression of the mouse C5aR1 in wild-type mouse. So we've been using this mouse model to induce acute lung injury, which is one of the features of what's happening in COVID-19 patients. And as you can see here, there is a dramatic ALI, acute lung injury, which is induced by C5 in this C5aR1 knockin mouse. You can appreciate the infiltration by CD45 positive cells. And these cells are basically cells that express LY6G and LY6C or LY6C only, so then neutrophils and monocytes. And this is associated with lung damage that can be seen by the release of albumin, a typical marker of lung damage. So now the obvious question is whether avdoralimab could actually do something. And in fact, it does. As you can see here, avdoralimab totally blocks acute lung injury, which mimics what's going on in COVID-19 patients, but here in this knockin mouse model. So more than that, now we've been going back to human cells and taking PBMCs and actually here isolated monocytes from COVID-19 patients. And you can see that the induction of IL-6, TNF, or CCL2 is blocked also by avdoralimab here in purple. So basically what I've been telling you today so far is the fact that C5a and C5aR1 may well play a key role in the initiation and maintenance of inflammatory response in COVID-19 patients, as we see an increase in soluble C5a, which is proportional to COVID-19 severity, and we do see high levels of C5aR1 in blood and pulmonary myeloid cells, and we'll see also that the anti-C5aR1 therapeutic antibodies, in particular avdoralimab, but not only, could prevent the C5a mediated activation, in particular, could inhibit acute lung injury in human C5aR1 knockin mice. So this is a model that we are proposing, actually, trying to compare non-severe COVID and severe COVID disease. So I think it becomes clearer and clearer that there are two phases, the epithelial disease and the so-called endothelial disease. So in this epithelial disease, the SARS-CoV-2 here is actually infecting the epithelial cells. You can recognize the pneumocytes, type 1 and type 2. And thanks to the action of the immune system, the complement cascade and other factors can be activated, and the viral load is actually restricted and maintained to low level and eliminated. But in severe COVID-19, for reasons which are unclear, SARS-CoV-2 can escape the immune system, can cross the epithelium, and then start to infect endothelial cells as well. In this condition, the innate immune system is overwhelmed, and there is a massive recruitment and activation of myeloid cells by C5a. And we do think, because the literature is really supporting that there is also a role of C5a in the endothelialitis and the microthrombosis, which are really seen in these COVID-19 patients. So this is where the Explore COVID translational study becomes a clinical trial and becomes FORCE, that stands for COVID elimination. And I'm almost finished, but I just want to share with you what is FORCE about. So FORCE is a phase 2 clinical trial with 2 cohorts of patients, and we don't have the result of this clinical trial as we are recruiting patients as I speak. We are recruiting patients of 2 categories. Patients with severe COVID-19 pneumonia, but who don't require hospitalization in ICU, or people with ARDS. And obviously, it's a double-blind randomized clinical trial comparing avdoralimab to and best supportive care to placebo and best supportive care, with the primary endpoint being precise here. So as I told you, this clinical trial is recruiting as I speak, but others, in particular, this is depicted here when Infiltration, another biotech company, actually published 5 days ago very encouraging results for an exploratory phase 2. And what they are doing here is to block C5a, not C5aR1, but they are very interesting data, which were actually following a very impressive case report published earlier during the year. So basically, I've been telling you already all that, but I would like also now to conclude by telling you that all these results support the fact that C5a-C5aR1 axis blockade could be a mean to limit myeloid cell infiltration in damaged organs and prevent the excessive lung inflammation and endothelialitis associated with ARDS in COVID-19 patients. And we do think that this is a good example of the possibility to reposition, to repurpose therapeutic blocking antibodies, such as the anti-C5aR1 avdoralimab, but others as well, as I've been showing you for the Infiltration. So I will stop here. I'm not over time as far as I see. I first want to thank patients and their families for participation to this translational study and the clinical trial. And I have a lot of people to thank. So people at Innate Pharma, in particular Vivier de Maria, who really led the group, but also Julien Carvelli and all our colleagues, MDs, who are involved in the clinical trial and who were involved in the exploratory study. And also Frédéric Velli, who is running the clinical lab for us, and all the other people from Marseille Immunopole and the COVID-19 group at Innate Pharma. And I will stop here, and I'm happy to take any questions. Thanks for your attention. Abdallah, we don't hear you. You might probably on mute.
How about now?
This
How
is
about
perfect.
Now? Good, good. I was saying, great presentation, Eric. Thank you very much. It was very informative, very rich. We learned a lot. And actually, I have a lot of questions. I don't know how we're going to deal with that, but I'm going to give you questions until you say stop.
Okay, go ahead, make my day.
Yeah. So, one first question, Eric, is regarding the lymphopenia.
Mhm.
One attendee was asking whether the decrease, do the blood lymphocyte number go down because they are recruited into the lung?
Okay, so we don't have a definitive answer to that. I'm not sure it's actually well known. There are a number of published papers which report as well this lymphopenia. As far as I understood, there is a combination of apoptosis of some subset of lymphoid cells, but also of infiltration at the inflammation site. I think it's a combination of both.
Okay, so another question is related actually to the level of C5. Where would you situate or locate C5 regarding the cytokine storm, for instance? So one question was about whether we could use the C5 axis as a warning actually against the
Mhm.
cytokine storm, and whether if you use this, the second actually linked question, if it's that's the case, then if you use, for instance, C5 as a target, would be no longer using or no longer needing to use an inhibitor of the cytokine storm if it's ahead, if it's earlier in the pathway.
That's a very good question. As you could see, I didn't claim that C5a was upstream of the cytokine storm. I was just saying that there are data supporting that it could be the case. But I think that it's likely, and this is my personal feeling, is that C5a is obviously not the only factor to initiate the cytokine storm, but it contributes to the cytokine storm. And it contributes to the cytokine storm in two ways. First, by inducing the migration of myeloid cells to the site of inflammation. This is obviously very important. The production of cytokines within the inflamed lung is a product of two factors: the number of cells that can produce the cytokines and the amount of cytokines produced by one given cell. So you have to multiply them to have the total explanation of the cytokine production. So for sure, C5a is a very potent chemoattractant molecule. And I've been showing you that both in vitro using neutrophils from healthy individuals, but also in the C5aR1 knockin mouse in the acute lung injury. So I do think that this is a way that C5a contributes. Now, whether C5a can be monitored in COVID-19 patients, yes, obviously. Okay, this is an important factor, and whether it's a warning factor, we still don't know that. We need many more patients to be fully convinced of that, and whether one can treat or not patients using anti-cytokines in the combination of doralimab or other blockers of the C5a, we just don't know. The clinical trials are actually made for that. They are made to answer to the question whether the blockade of the C5a, C5aR1 axis can be of interest for the patients. And as I've been telling you 5 days ago, there are very interesting data showing monotherapy results with an anti-C5a blockers.
Great. Eric, do you think that C5 targeting would help with dexamethasone?
Okay,
Do you
so
think?
that's a very good question. Yeah, again, I'm not some kind of prophet. What I'm trying to do is to translate the data from basic science into possibility of treatment for the patients through clinical trials. So as long as the clinical trials are not held, I would certainly not tell you anything on the results. But what I can tell you is that it makes a lot of sense. Now that we know that the blockade of the C5a pathway as some interesting signal in this phase 2 clinical trial from our colleagues at Inflarix, it makes sense indeed to think of possibilities of developing clinical trials in association with other molecules. And as the UK group released information for the role of corticoids in COVID-19 patients, why not this kind of molecule, obviously?
Another question is related to whether you have checked the C5a, C5aR1 axis after recovery.
I'm sorry, I don't hear you very well.
Yeah. Yeah, so
Yeah, much
another
better.
question, yeah, another question is related to whether you have checked the C5a, C5aR1 axis after the recovery of patients.
No, we didn't do that. We didn't do that. It's a good question. I don't have the results for that, unfortunately. I don't expect to see a major difference in C5aR1. As I told you, C5aR1 is really expressed on 100% of neutrophils and circulating monocytes. It's expressed as steady states. It's not the limiting factor. The limiting factor is C5a itself. So we don't know for the patients after recovery. All I can tell you is that C5a is not detectable in healthy individuals. So my expectation would be that if we would look at C5a and C5aR1 in patients after recovery, C5a would be down and C5aR1 would be still very high. Because again,
Okay.
for C5a to be able to do something, it needs to recognize, for C5aR1 to do something, it needs obviously to interact with C5a. And C5a is the limiting factor.
And you think that's the when you look at the expression actually of the C5a receptor, it's almost always high, right?
Yes, it is.
Right, in contrast to C5a, which would go up,
Yes,
with the severity of the disease. So that
yes.
means that's even. So how do you explain that actually, Eric?
Well, as I told you, I mean, C5aR1 is expressed at steady state on all myeloid cells. Okay?
Yeah.
But it doesn't do anything unless it encounters C5a. So C5a is induced during inflammation. It can be actually induced here directly by the virus, by this N protein of coronavirus, but it also can be induced by CRP, by the way. It can be also induced by obviously antibodies. This is a classical pathway of complement activation. So there are different pathways that could lead to C5a activation in COVID-19 patients, and this is a limiting factor.
Yeah, another question. You let me know if you're tired, Eric.
No, no, I'm fine. I'm fine.
is there is there an age difference in terms of the expression of C5a
Uh,
and the role
Again, I
eventually?
was probably not clear enough. What is the key difference for C5a is the level of inflammation. If you have inflammation, if you have infection, you can have C5a. I mean, this is well known for a lot of chronic inflammatory diseases, such as vasculitis and other inflammatory diseases. This is a case for this coronavirus infection. So we didn't look at kids in particular. Whether C5a could be also present in kids upon SARS-CoV-2 infection, if this is a question. Uh,
Okay. Another question is related to the type of macrophages which are prevalent in the COVID-19 patients. Do you have an idea? The
no, so
type
we
of
didn't look at this
macrophages.
in detail. It's a very good question. Other groups have done that and are doing that. So I'm not going to say more than what I've said. We say expression of classical myeloid cell markers, CD163, CD68, and a substantial fraction of them co-express the C5aR1 in the lung. That's what we can see. But no, others will do that or have done that already.
Okay. I two more questions and we will stop there. So
It's up to
one
you. I
is,
can go
yeah.
for the entire day.
A question is related to IL-8. I'm not sure if whether an attendee asked about IL-8. What was the level of IL-8 in the bloodstream? Did you look at that?
Yeah. So we did look at that. And actually, IL-8 was particularly very high in the broncho-alveolar lavage fluid. I've been showing you the result of that, which is extremely important because if you have IL-8 in the inflamed lung, you can have a rational or a mechanism, a potential mechanism for the infiltration of neutrophils in the inflamed lung. So yes, we did see IL-8, CXCL8.
Okay. You looked mainly at the antibodies, I mean, blocking antibody against C5aR1.
Hmm?
I'm wondering whether you think would I mean targeting C5a C5aR1 is better than targeting C5a, the soluble factor? I mean, the ligand?
No, I'm not saying that. I'm not saying
No,
that it's better. Again, I mean, only the clinical trial will tell us. The clinical trial, the phase 2 clinical trial addressing the role of C5a in COVID-19 patients has just been released. Now, those colleagues are thinking of going into phase 3. We are still recruiting patients in phase 2. So once we have the result of phase 2, we will compare the results. It's definitely the same pathway. Now, whether pharmacology speaking, or the saturation of the molecule, saturation of the receptor, saturation of the ligand might have differences in terms of efficacy. I just don't know. But you mentioned also that we were using only antibodies. That's true. Although in vitro, we have been using also avacopan, which is a small molecule that blocks C5aR1. And at least in vitro, we could see that there are comparable action of C5aR1 antibodies, have C5aR1 antagonist, and anti-C5a itself. So
yeah, actually, another, I guess, last question is related. Usually, when we talk about COVID-19, we talk about a viral stage or viral phase where we can use antiviral molecules, and then an inflammatory phase where we can use basically anti-inflammatory molecules. And although you haven't stressed it much here in terms of immune checkpoint, where you can basically release the immune system to do its job, where would you locate that? Is it in between both phases?
okay, so antiviral drug would be very nice if they were very effective, which I think it can be disputed at present. As for immune checkpoints, yes, we have studied them, in particular in lymphoid cells. We have been paying attention to PD-1, to CD39, to NKG2A, and as I mentioned very briefly in the introduction, in the interest of time, I didn't go through the result, but this is in press in cellular and molecular immunology. So we've seen an increase in NKG2A, we've seen an increase in CD39, an increase in PD-1 on NK cells. We could actually document an increase of NKG2A as well on CD8 T cells. So there is an idea that one can actually unleash these immune effector cells, because we know that NK cells and CD8 T cells are so important in the control of virus infection. So it will make some sense actually to treat very early on during the disease patients with immune checkpoint inhibitors to harness the antiviral function of these immune effectors, and then maybe a combination of anti-C5aR1 or anti-C5a or blockers of the C5a-C5aR1 pathway, so that one can diminish the likelihood of having inflammatory diseases. But this is something, again, that the clinical trials will tell us.
Sure. Very interesting. Thank you very much, Eric. I think we will stop here.
Well, thank you.
Very
thank
much.
you all.
Very informative, very rich. Thank you very much to all attendees. And I think we will stop here. Thank you very much to all.
Okay, thank you very much to everybody. And you can contact me by email if you want more information.
Okay, thank you very much.